human gas exchange and spirometers. So if we start with inspiration and expiration, so that's part of ventilation. Remember, gas exchange is about diffusion across cells, but ventilation is mass transport of gases in and out, and that's all to do with pressure gradients. So what you have to do in order to get the air to move into the lungs is you have to make the volume inside the lungs much bigger. And you do that by contracting the diaphragm muscle. So normally the diaphragm muscle is a dome shape. When you contract it, it flattens. And the external intercostal muscles, intercostal just because they're between the ribs, external because they sit on the outside, they hold on to each other, and as they contract they raise the rib cage up and out, so the total volume is increased. And if the volume increases, then the pressure decreases, and the pressure inside the lung cavity is then smaller than the pressure that's outside, so air moves in down a pressure gradient through the trachea, through the bronchi, through the bronchioles, into the alveoli. The alveoli stretch because they're full of elastic fibres. Next, if you want to breathe out, you have to make the volume inside the lungs smaller. So to do that, we need to relax everything. So because there's lots of elastic fibres in your lung tissue, that will just recoil as soon as the external intercostal muscles relax. So the external muscles relax, the diaphragm relaxes, and the elastic recoil of the alveoli causes the volume to then decrease. So the diaphragm becomes a dome shape again, the muscles relax, the external intercostal muscles relax, that allows the lungs to recoil, and the volume gets smaller and the pressure gets greater than outside in the atmosphere, so the air moves out of the alveoli, into the bronchioles, into the bronchi, into the trachea, and out into the outside atmosphere. You can also mention the contraction of the internal intercostal muscles. And that will help to make the volume of the lungs decrease faster so that you can force more air out more quickly, for example in forced expiration. But the most important point to remember is its relaxation of the external intercostal muscles and the diaphragm muscle, as well as elastic recoil or internal intercostal muscles contracting, and it's those things together that make the volume decrease, the pressure increase, and air moves out down a pressure gradient. So that's the movement of air in and out of the lungs. That's called ventilation. Now, as part of ventilation, the main reason for ventilating is to maintain a concentration gradient across the alveoli walls. So if we take the alveoli, they are a cloud shape which increases the surface area. They have the capillaries running very, very close. The capillaries are very narrow, such that the red blood cells are squeezed through, so that slows the rate of flow. And all of that helps to keep the diffusion distance as short as possible, the surface area as big as possible, diffusion pathway short. So if you are asked how the lungs are adapted, you would say that the alveoli are folded, that increases the surface area. There are many, many capillaries all over the many, many alveoli, that will increase the surface area. The red blood cells are squeezed so that they're flat against the wall of the capillary, that increases the surface area of the exposed for the gas exchange. The diffusion pathway is kept short, that's because the capillary wall is one cell thick, and that one cell, that endothelial cell, is very, very thin. The epithelial cell in the alveoli wall is very, very thin, so epithelial cells are thin. And so we have a really, really short diffusion pathway from the inside of the alveolus into the red blood cell. Is only 2 cells thin, and those cells are really, really thin. So that's a really short diffusion pathway. And then the final thing is the concentration gradient. So the concentration gradient is maintained because of ventilation. So the more you ventilate, the more you refresh the oxygen, and then the oxygen will diffuse down its concentration gradient into the red blood cells. But if those red blood cells stayed put, that wouldn't be very helpful, so we need blood flow, and that's maintained by the contraction of the right ventricle, and that helps to pump the blood through the pulmonary system, maintaining a blood flow. So as each red blood cell picks up oxygen, it's moved along the capillary, and that allows for more oxygen to diffuse down its concentration gradient. So that's ventilation and maintenance of concentration gradients for humans. All of that is for the role of oxygen. So you're trying to get oxygen into the blood because all cells need oxygen for aerobic respiration. And again, that's on the cellular level. Don't confuse respiration with ventilation. Ventilation is the mechanism behind breathing and getting fresh oxygen air into the lungs and out of the lungs. Aerobic respiration is what happens at a cellular level with that oxygen. Now, the other kind of questions that you get about lungs and breathing focus on the graphs that you get. So you would usually have a time along the x-axis and then either lung volume or spirometer trace. A spirometer is just a piece of equipment that we use to measure lung volume, and what we will do with a spirometer trace is usually you have a barrel and a box of water, and this is connected with a pen that records on a drum. So that means that you are connected to the spirometer via a tube, so this is a person breathing in and out of the spirometer. As you breathe out, that causes the drum to go up, that causes the pen to go up, and you get an upward curve. As you breathe in, then the drum goes down and you get downward. So if you are given a trace like this, and it said it's a spirometer trace, then you would have to say which is the inhalation, which is the exhalation. Well, exhalation pushes the drum up, pushes the pen up, so this is exhaling, this is inhaling. In contrast, if it's a graph to show lung volume, then obviously when you are breathing in, your lung volume is getting bigger. So in that case, a trace, if it's just lung volume, would be inhaling and exhaling. So just be careful of that. Be mindful what it says on the y-axis. In terms of other labels, for example, what does this show? Well, if it's a normal breath in or out, it's tidal volume. So you would label that as tidal volume. If you were asked to measure the tidal volume, you would just go peak to trough and average that out over at least 3. So the same would go for both spirometer trace and the lung volume trace. Same mechanism for the calculations. The other thing that you might be asked to measure is your vital capacity. So your vital capacity is the total volume of air breathed in and out in one full breath. Remember, it's in and out for vital, in or out for tidal. You do those measurements. If you have the time, on the x-axis you can also work out the breathing rate. So if you know that you're starting from this point, that's 1 repeat, that's 2 repeats, that's 3 repeats. So in that time frame, you've had 3 breaths. So then you can work out the breathing rate by saying 3 breaths took this amount of time, therefore it's 1 breath per however many seconds that works out to. Or if you need to work it out for a minute, then you scale it up for the minute. The other thing that they might ask you is to do oxygen consumption. Now that is done on a spirometer trace, and that's because on a spirometer trace, because it would be poisonous to keep breathing in your own carbon dioxide that you've just expelled into that machine, what the machine does, or what it has on it, is a scavenger. So before the tube gets to your mouth, it has a carbon dioxide scavenger in that box, like soda lime or something like that, that can absorb the carbon dioxide. So the volume that goes back into the box is actually less than the volume that came out. And what that will do to the trace is you might start somewhere up here, but over time, the trace will come down. And so what you can do from that is if you know what it's starting at, so you work out the volume here, you work out the volume there, you work out the time that it started at, which is from that point there to this point here. So you work out your time, you work out the change in volume, And that change in volume equals the oxygen consumed. So you can work out oxygen consumption over time, which is the rate of oxygen consumption. You could also, from that graph, work out the breathing rate as well, because you would know the number of peaks you've got indicates the number of breaths given in a particular time. That gives you the breathing rate as well. So those are the typical kind of things that you get on spirometer traces and gas exchange systems.